Next up, we have Dr. Kai-Shu Ling talking about CGMMV and WGMMV.
Thanks to my colleagues that gave a lot of good background, so making my job here a little bit easier. And I'm just so happy to be the last person in this whole series of the seminar. So thank you for all of you kind of stay on for this webinar. And so my name is Kai Ling. I'm a research plant pathologist with USDA AIs in Charleston, South Carolina. So here's my email if you want to contact. So my focus here on this is on the seed and mechanically transmitted viruses on using this cucumber green mottle mosaic virus and watermelon green mottle mosaic viruses. So they kind of use CGMMV and then WGMMV. Those are both tobacco viruses. And so here's kind of showing some of the symptoms and early detections in reporting in the North America. This is on the cucumbers in Canada in the early on when our lab was working there and helping out identify this CGMMV in the greenhouse on cucumber, and then in the California, really the same year that in California they identified that in the on melon and watermelon, and not just symptoms on the mosaic, but also have impact on these the fruits on the so that is where the it can be a serious problem because of the affecting these more like spongy flesh, and it can be rotting, and then there'll be total crop lost. So kind of important on that. And so this is kind of the taken from the distribution map in the world. So you can see this pretty much quite widespread, many in the Europe and Asia, but the most recent one in the North America and Australia, and most of the recent one is actually in Africa, in Nigeria, and then so this is where can be quite widespread in the world. So why it's these global outbreaks? So this is kind of the cucumber green mottle mosaic virus. It's kind of similar to other tobacco. More famous one would be tobacco mosaic virus. So it was first discovered in Europe in 1935, and then it's still the province over there. The outbreak kind of spreading out to many Asian countries. And then first report in North America, as I mentioned, it was about 2013, 2014 time in California and also in Canada. And most recent one in Australia and also in Nigeria. So for the genetic diversity on the CGMMV isolates, they usually kind of break it into two genotypes, one Asia and one Europeans. Those are the major ones. Some of the kind of in the between, but most of those are quite of the kind of dip into these Asian and then European the genotypes. The complication of the detection is that there are at least 6 these tobacco viruses infecting cucurbit crops. So this is kind of making it complicated in a way that for diagnostics, since there are so many been reported on that, and then I will be focusing just more on the cucumber green mottle mosaic virus, and then the watermelon green mottle mosaic virus. But there's other viruses like cucumber mottle virus, Cucurbit green mottle mosaic virus, zucchini green mottle mosaic virus, and cucumber fruit mottle mosaic virus. So those are kind of several of the very similar, but the sequence are quite different among those, some kind of relationship. But the watermelon green mottle mosaic viruses, this is actually one of the newest one on this tobacco virus series. And using this reporting was 2019 from Taiwan and also from California. The sample that used in Taiwan was from collecting for 2007, and then in California was 2017. So there's no serological, this watermelon green mottle mosaic virus has no serological relationship with CGMMV, which means that when using ELISA for testing for CGMMV for detection or for seed health testing, you actually going to miss this watermelon green mottle mosaic virus. They are sharing about 62 to 81% of the nucleotide sequence identity to other tobacco viruses. So you can actually designing these primers can specifically detecting those viruses. So why this the CGMMV accelerating its global distribution in recent years? So oftentimes it's offshore this hybrid seed production in Asia countries where this CGMMV is widespread. So increasing this global seed trade to many other countries so around the world, so there can be the potential help to kind of spreading out to other regions. So increasing production of the cucurbit under the protected environments. So not just in the field production, but CGMMV oftentimes in the greenhouse, in the food house, and then those kind of to the plant handling, the mechanical transmission, and grafting this cucurbit to loose stalks where the CGMMV can spread quickly in the nursery propagation. So there's three seed-borne viruses on cucurbits that earlier colleagues mentioned about the melon necrotic spot virus and CGMMV, and then there's a squash mosaic virus. They kind of those are the one that three of those seed-borne. We already see here some more new ones, but those are the one that kind of more traditional seed-borne viruses that are used for the seed health testing. The seed transmission of these CGMMV is a seed-borne, which is easy mechanical transmission. So new infection can initiate from contaminated seed to seedlings. And seed health testing and seed treatments are necessary for the management, hygiene, and then disinfections are practical measures for cucurbit crop production. During the production, also need to be make sure that you are actually not transmitting those viruses. So no cultivar currently are resistant to CGMMV, but there's some tolerance in the cucumber. There's very high seed contamination in some of the seeds, but there's low seed transmission actually. So there's seed-borne doesn't necessarily mean seed transmission. So I had to be kind of careful about how, but there's understanding that the mechanical mechanism of the seed transmissibility is that when through seeding grow out with contaminated seeds really low or do no transmission, but when the bioassay using this mechanical transmission using these contaminated seeds, it can actually generating high rates of transmission to the seedlings. So the seed treatments, one of the methods that used to managing the CGMMV is to the goal is to develop this effective seed treatment through the thermal, by heat, or by chemotherapy like chemicals that without ever effect on the seed germination. So typically the dry heat treatments using 70 to 72 degrees for 3 days, about 72 hours, and chemical treatments typically using 10% TSP trisodium phosphate, soaking seeds for 30 minutes. Combining these two treatments, usually giving a better of results, can achieve by using the TSP treatment for soaking for 30 minutes, and then using the heat treatment for 72 hours. So the typical symptoms, it can actually infecting many of the cucurbits. Here just kind of showing some of the typical symptoms on the different melons, bottle galls, cucumbers, watermelon, and squash. So it can be, and to prevent this introduction through this seed health testing, and usually using the serological like ELISA or molecular methods. So purchase those seeds and transplant from sources that test for CGMMV. Higher risk when using these untested seeds. Some growers are using more like a self-save those seeds. Those are typically more risk using those seeds. And preventing introduction using disinfectants on facility, equipment, tools, or clothing, and then the hands. And in the field, not need to be more monitor about the symptoms, and then using some of the field-based testing like immunostrip. Rotation with non-host crop for kind of quite long. It had 2-3 years because tobacco virus is very persistent in the soil. Eliminating these cucurbit seeds, wheat, and then the volunteer plants. So you had some of the cucurbit wheats the host. And not all the disinfectants are creating echoes, so you have to be selecting the right disinfectants. Using this is one of our experiment was screening some of the about 20 some of the chemicals and showing these products good, and then mentioning about TSP very quite effective. And what we have identified some of the voricides actually are very good, and that we're actually recommending also in tomatoes crops for tomato brown local fruit rots. So those are some of those need to be selecting. As I mentioned, there's no resistance in watermelons area, so we're actually screening some of the watermelon germplasm for resistance, and most of them are really there's no resistance, but some tolerance one. We identify some of those from the germplasm that has a tolerance to CGMMV. It's actually more wide collagen, so it's the but still generating some of the population, breeding population, still kind of working on trying to figure out what the genetic inheritance of resistance and the market development. So in summary, so you need those the viruses I mentioned about CGMMV and watermelon, the green mottle mosaic viruses are in the United States are the control and the official control. So the quarantine inspection test is kind of important. Planting the virus-free seeds using the seed health testing. Seed treatments can be used to reduce the virus in the contaminated seeds. And hygiene practice, choosing the proper disinfectants for disinfection. Nursery seeding, germination, grafting plants need to be careful monitoring and then testing and using disinfectants, scouting and disease diagnostics in the nursery and the field, logging those disease plant and early plants, and rotation to non-host crops, and removing weed of potential virus reservoir and breeding for disease resistance, which is still not yet have the for watermelon has no resistance source at this no resistant cultivars at this moment. So I wanted to thank to my group. Those are just kind of some of the people actually working in the lab early on, but the funding agencies and supporting this work. Thank you.